Okay. Right. So today's subject is DNA replication and the genetic code. And if you remember where we left off last time, we had these three questions. What is DNA, which we covered since its first discovery way back when in the 1800s. How do we know DNA is the genetic material? We covered the history of experiments that sort of led to the final proof that DNA is the genetic material. We talked about how everybody thought it was going to be protein, but it turned out that DNA is actually the stuff of our genes. And then we talked a little bit about, at the end, about how the structure of DNA, which was discovered by Watson and Crick, implied a potential mechanism whereby the copying of the DNA could happen, and that is the inheritance of this genetic material could go on from generation to generation. And so what we're going to start out with today is picking up on that question and really learning a lot more about how DNA is copied. And then the questions we're going to deal with today go on from there to what is a gene? I sort of skirted that question last time, if you remember. I said, you probably know enough about genes where I can just sort of use the word gene for a while and not really define it yet. So today is when we're really going to start to define what a gene is. And we're going to talk about how the DNA actually carries information. You know, how does it work as an informational hereditary molecule? How is that information read? And then this last question is of a different kind than the first three questions, obviously. How much of who we are is in our DNA is a huge, huge question with many philosophical and sociological and every other kind of implication. And it's not a question we're going to ask and answer today, but it's the kind of question I want you to start thinking about throughout the course. Because the more you learn about DNA and genes and genomes, the better you should be able to think about this question as it impacts, you know, many aspects of your lives. Okay, so this is where we left off last time. Watson and Crick had just discovered this double helical shape of DNA, which you see there. And in their paper reporting this structure, they made the claim that just by looking at this structure, this double helix winding around itself, it implies how it might be copied as cells divide. And as we talked about last time, the hypothesis, that is their educated guess about how this works, is that because you have two strands that have exactly the same information in them, that is, if you know that this one is ATGC, you know that this one is TACG. There's that complementarity between T's and A's and G's and C's. And so if you know one strand, then you can write down what the other strand is. That implies that each strand could then serve as a template to be copied from, and then you'd get two copies of the original thing, and it would be identical copies. So that's what they thought. But then a very important experiment had to be done to actually show that that was the case, and it did turn out to be true. And this experiment is called now the Mendelson-Stoll experiment, after the two people who did it. This, again, is one of those classic experiments in biology that every sort of undergraduate biology major has to learn at some point, and then they probably hear about it again in graduate school. It's in a way the epitome of an elegant experiment. And moreover, it's a great example of sort of creativity in science, because not only did they conceive of a very simple, elegant experiment, but they actually invented a technology to answer that question. Okay, so as you'll see through the course, we'll be learning about a lot of examples where knowledge is really pushed forward by creative people coming up with new ways of looking at the world and doing experiments. And this is one of those examples from now quite a long time ago that really epitomizes how a creative mind can think about a problem and devise a way of answering that problem. So here's the question, right? So Watson and Crick say, well, the probable way that DNA is copied is that you have this double helix, okay? And then you somehow separate the two strands, right? And then each one of them is copied. And the copying is sort of obvious because everywhere there's an A, you put a T. Everywhere there's a G, you put a C, okay? And so that's what's represented here, where the strands that are getting copied are in red, and then the new strands are in pink, okay? So the result of copying this molecule then is each new molecule has one old strand and one new strand. Does that make sense? Okay? And Mendelson and Stoll called this the semi-conservative mechanism or hypothesis. Semi-conservative because you're conserving both of the original strands, but in each of the new copies, half of it is old and half of it is new. It could also have been true that that was not how it worked, but instead how it worked is by what they call the conservative mechanism, where DNA just happens to be a double-stranded molecule. And so you have this double-stranded molecule, and then when copying takes place, you just make a new one. And somehow this new one is an identical copy of the old one. Okay? And you can see how these give sort of a very different result, where in this case, each of the new copies is half old and half new. And in this case, you have one version that's still the old one and one version that's completely new. And then they went even beyond that and said, well, okay, there might even be a trickier possibility that would be that it's sort of conservative, but the cell doesn't do it all at once. It doesn't just make one long copy of this and turn it into this, but instead it does it in pieces. And so you get a big chunk of old, and then its copy that's new. But then another part of this long chromosome, remember chromosomes are, in you are many millions of these ladder steps long. Okay, so maybe it does it in chunks. Maybe this chunk you have old and you make a new copy, but then that new copy is attached to what used to be the old copy, and then you get a new copy up here of that. And so you still get two molecules in the end, okay? But each one is part old, part new, okay? So these are the three possibilities they laid out, and this is an important step in doing science. It's what I'll often refer to in the class as developing a model, okay, or a set of models. So a model in science is basically a representation of your understanding or your postulation of how a biological process happens, okay? So their model that they favored, that they thought was going to be true, is a semi-conservative model, where the original molecule is split in two, and then each one gets a new strand copied off of it. And then there are alternative models, the conservative model and the dispersive model, and the idea behind doing an experiment is to distinguish between those models. And then you go from a state where you had a model to one where you have the truth, where you have an answer. Okay, so how did they... So their task, right, is to distinguish between these possibilities somehow. And that's tricky because you can't just look at DNA. If I gave you some DNA, you wouldn't be able to say, oh, that's old or oh, that's new, right? So they had to figure out a way to do that. And they devised this technology to do that we're going to go through on the next few slides. Here's the experiment that they did. They worked with bacteria, okay? The advantage of bacteria is that they divide every 20 minutes. Okay, they're very fast growing. You can get large numbers of them in a very short amount of time. Okay, that's the advantage in the lab. The advantage out in the world is on the bacteria side. Because they have that ability to grow so fast, they make you sick. Okay, so in the lab, bacteria are very convenient to use because they grow so fast. Okay, and so what they did was they grew the bacteria on their favorite medium, which is basically chicken soup. And then what they did special, though, was instead of using regular nitrogen, remember DNA has a lot of nitrogen in it, they used what's called heavy nitrogen, or nitrogen 15. Okay? And so just for those of you who need a quick refresher, this is not a chemistry class, so don't get worried, right? So this is the periodic table of the elements. Okay? Nitrogen is right here. It has an atomic weight of 14 because it has 7 neutrons and 7 protons. Okay, any of this sound familiar? Okay, right. So that's normal nitrogen, atomic weight 14. Okay, but there are versions of nitrogen that have an extra neutron, say, and that's nitrogen 15 because it then has 8 neutrons, 7 protons, and so it has an extra unit of atomic mass. What's nice about that is that then it is indeed heavier than regular nitrogen, okay? All of those little neutrons add up, okay? And so if you have a method that can basically weigh those differences, you can detect whether you have heavy nitrogen or light nitrogen in whatever molecule you're studying. And so they use that to their advantage to devise this really clever experiment. Okay, so what they did was they grew the bacteria in heavy nitrogen nucleotides. So that is all of the A's, C's, G's, and T's that the bacteria were growing on had nitrogen 15. Then what they did was they washed the cells so that all of these went away, and they replaced it with normal nitrogen medium, okay, normal A's, C's, G's, and T's. That's what we'll call light nitrogen. And then they let the bacteria grow for exactly 1 generation, so exactly 20 minutes. Okay? So basically what they've done is they've made all of the bacterial DNA before this step heavy, right? And then they go 1 more generation and allow the cells to incorporate light nitrogen. And now if they can tell the difference between the heavy and the light, that will allow them to start distinguishing these models, because it's the weight of the nitrogen that tells you if you're old or new. Okay? So how do they do that? Well, they go through this step, grow them heavy, add light, let them go 1 more generation, and then they take all the bacteria and they extract the DNA from them, okay? And you'll get a better handle on what that means next week in lab. And then they use this new technology they invented called high-speed ultracentrifugation to separate the DNA by density. Okay? And so here's a schematic of what that looks like. Okay? So basically what you can imagine is a test tube or a slightly larger thing that's filled with a solution where the light stuff is on top and the heavy stuff is on the bottom. Okay? And so that's the normal course of events, right? If you have something that's less dense, it'll float on top of something that's more dense, right? Okay? And so beyond that, what will also happen is that if you place something in a density gradient like this, where you go from light to heavy, it will settle out to wherever its density is. Okay? So for example, if you go into a swimming pool, your tendency is to sort of be buoyant because most of your body is water. Okay? If you go into the Dead Sea, which is very, very heavy in salt and concentrated, it's more dense than normal water, you float very easily. You basically sit on top of the water. Okay? So imagine that you can have DNA sort of find its level in a density gradient, and the light nitrogen will sit up here, and the heavy nitrogen will sit down here. Okay, does that make sense? Okay, it's going to make more sense now when I do this experiment, as long as it works. Okay, so I have 2 solutions here, very special chemicals. Okay, so I'm going to add this one to my graduated cylinder, right? And then I'm going to add this one very carefully. Try not to make too much of a disturbance. Hopefully it'll work. Yeah, okay. So the people in the front row can vouch for me. What's the color at the very bottom? Clear. And what's the color at the very top? Not clear. Okay, so we have a brown liquid up here and a colorless liquid down here. Okay, and that's because the colorless liquid is very dense. Okay, and the brown liquid is not dense, or is less dense. Okay, and what you can see if you look really closely, I'm going to walk around a little so you all see my wonderful experiment, right, is that there's a gradient. Okay, so it's darkest at the very top, and they've sort of mixed here so you get this light brown color. Okay, and then at the very bottom there really is no color whatsoever. Okay, does everyone believe me? Do you want me to come over there, or do you believe me? Do you want to see it? All right. So that's basically this density gradient, and if I put something in that was sort of the density in between these two things, it would sort of float in the middle, right? If I put something in that was very low density, it would float on top, and if I put something in like lead, it would just sink right to the bottom. Okay? So these are the 2 chemicals I used. Okay, we have Sprite on the bottom and Diet Pepsi on top. Okay, and the reason the Sprite is dense is because it's regular Sprite and not Diet Sprite. Okay, sugar adds density to this liquid. This has NutraSweet in it. Okay, NutraSweet is so much sweeter than regular sugar that you only add a tiny bit. So this solution is not very dense. This solution is very dense. Now you have a party trick that you can show all your friends. And you now all understand what a density gradient is. Okay, so what did Mendelson and Stoll do with their density gradient? Well, they had this DNA that started out heavy, and they let the cells go one more generation on light. And so their expected result, if they were right, if semi-conservative replication was how DNA was copied, then in that first generation, all the molecules will be half old and half new, right? And if that molecule is half old and half new, then its density is halfway in between heavy and light. And so on the density gradient, it should fall out right in the middle. Okay? Now, if it were conservative instead of semi-conservative, then you'd have one completely old molecule and one completely new molecule each time a cell divided, right? And so what would that look like here? Try not to look at the next slide on your notes.
One at the way top and one at the way bottom.
Right. So you'd have your old DNA at the bottom, and then your new copy would be at the top. And what if it were, again, don't look at your next slide in your notes, what if it were dispersive? That is, if you copied chunks of double-stranded old and double-stranded new, but they sort of were each attached to each other randomly along the same chromosome. What would you get?
Well, if it was equal, like if it was an equal amount, then wouldn't you get some form of this, but just not like one?
Yeah, so you're on the right track, right? You brought up an important parameter, right, which is how equal it is, right? How equal those pieces are, okay? So imagine that it's random and that these chromosomes are really, really, really long. Okay, so you're going old, new, old, new, old, new, and there's basically the equal probability of being old or new. So if you average over the whole length of the chromosome, what is the density on average? It's about in the middle. And it might indeed be spread out a little bit because there's that randomness involved, okay? But in general, the expectation is that, again, it would fall somewhere in the middle because each long, long DNA molecule is sort of half old, half new. Okay, so this leads to a problem, right? So we have three models. We have semi-conservative, conservative, and dispersive, and two of them give the same prediction, right? Now, just doing this experiment, though, you could rule out conservative, right? If you got this result, right, you did your experiment, all the DNA fell out in the middle, then just from that experiment, you could say it's not conservative. It's either semi-conservative or dispersive. Okay, so fine. So, and that's exactly what happened. They got this result, and then they said, okay, well, now we know it's either semi-conservative or dispersive. It's not conservative. So now we have to figure out how to tell semi-conservative and dispersive apart. Okay, so how did they do that? Well, they used their same method. Okay, and this slide is going to look exactly the same as the first one, except for one important change. So again, they grow the bacteria for a long time on the heavy nitrogen. That means all of the old DNA is heavy. And then, again, they wash the cells and they replace all the medium with medium containing the light nitrogen. Okay? But this time, instead of growing for one generation, they grow it for 2 generations. Okay? Then they do the same thing. They extract the DNA, they run it on their density gradient, and they see where it falls. Okay, so now let's think about this. What's the prediction if it's semi-conservative for this experiment? Okay, we already know that after one generation, every molecule for semi-conservative contains half heavy and half light, right? So what happens if you let the bacteria copy their DNA one more time? Right, so these two are going to separate, right? So you have a old heavy and a light new, and then each is going to get copied, but it's all light, right? So now what you have is an old and a new over here, and a new over here, okay? Light, light, heavy, light. Where does light, light go on this gradient? Very top. Where does heavy light go? Middle, okay? So this is the expected result for semi-conservative after 2 generations. half the DNA up here and half the DNA in the middle. What's the expectation for dispersive? Okay, this is a little bit trickier. For dispersive, you had these random sections of the same molecule that were sort of all heavy, all light, all heavy, all light. What happens if you let that process happen again? Take 30 seconds to think about it. Draw it out. Okay, this is sort of this is what these guys had to do before they did their experiment, right? Draw out what the potential result is, because if the potential result is the same as this, it's not a good experiment. Okay, so just start out and draw what it was at the end of the first generation, which is that you had, I'm not going to draw because you're not going to see it. Oops. You had something like this, right? Old, new, and so on. And then the other copy was new, old, and so on. Okay? So take this and go one more generation, assuming that it's still the dispersive model. And do it. Take 30 seconds and try and draw it out. All right. People have an answer? You want me to go through my answer? Okay. It would help to have three colors, right? So you have your old and your new, and then let's think about what the new would be, okay? Basically what you do is now imagine that this is all now one color. It's sort of reddish, right? And so the new molecule you want to draw is going to take random pieces of this and intersperse chunks of new. So let's say we had a blue color. Okay, so you take random chunks of this, and in between them you're now putting, say, blue. Okay, but it's not really three colors because the blue is actually the same density as the pink. Right? So if we then add it up, before we started, we have one molecule that's half red and half pink. Okay, and then after we go through the copying, now half of the new molecule is going to be half red and half pink, and then the other half of the new molecule is going to be blue. Does everyone sort of follow that? If you want, I can do it on the board. Does anyone want me to do it on the board? Yes, okay. Can you see if I just leave it here, or should I? I don't think so. Unfortunately, there's one color. Okay, I'm going to use dotted and dashed lines, right? So the old and the new were mixed in the first generation. Right? Everyone follow that? And we have sort of a really long molecule that's half solid and half dashed. Okay? And then to copy this, the way you have to think about it is, well, now let's take a random chunk of this. So let's take from, say, here to here. Okay? And that'll still be the way it was in the previous generation. Okay? But then my next chunk of that is going to be entirely new. Okay? And so entirely new, let's draw like that. Okay, sorry I don't have another color with me. Okay, and then it would go back to what it was in the last generation, right? And so on, and you'd be interspersing this, which now is sort of old, all of it, with really new stuff. Okay? But the point I was making is that every part of this molecule that is in the solid lines or in the red lines there is old, so it had the heavy nitrogen. And every part of it that is either dashed or these dots came when it was light nitrogen. Right? Because we're adding light nitrogen and then go 2 generations. So all of this is light nitrogen. And so if you average over this really, really, really long molecule, okay, half of it is going to be half heavy and half light. That was sort of the first generation, now old part of it. And half of it is going to be all light, because that's the new stuff, the new stuff. Okay? So if you add these all up together, what's the total density? It's 3/4 light. Okay, so that's the prediction for semi-conservative, and that's the prediction for dispersive. Okay, everyone with me? Everyone follow? Okay. Now, so that's the beauty of the experiment, right? That now, just by doing those 2 experiments, the 1-generation experiment and the 2-generation experiment, they can figure out exactly which model is correct. Okay? And so the actual result was that after 1 generation, all of the DNA was in the middle. And after 2 generations, half of it was up here and half of it was in the middle. And they had these distinct bands, and it was not lying at sort of 3/4 high. Okay? And so what that means is that the true answer is indeed semi-conservative copying of DNA. That is, the way DNA is copied is the 2 strands each serve as a template for copying a new strand, and so you get 2 identical molecules. Okay? I'm going to move this so you guys aren't blinded anymore. Okay, so beautiful experiment. On your DVD is an interview with Matt Mendelson, and he goes through a little bit sort of how the experiment was done in his own words. When you see the clip, you'll sort of fall in love with him. He looks like sort of your favorite uncle kind of guy. When I was in graduate school, he was the guy who taught genetics, and I was lucky enough to be his TA for genetics one year, and he's as sweet as he appears on the DVD. And he also is a really, really amazing guy, because in addition to being a great scientist, he spent many, many years fighting to limit the spread of chemical and biological weapons. And so he's sort of has a big heart in both sort of the personal sense and in the big world sense. Okay. Now, we know now, after Mendelson and Stoll, that DNA is copied semi-conservatively. Okay? Now, we need to get into a little bit more detail about how that's done, because this will become important in terms of how those letters are actually turned into information in the cell. Okay? And so, if you remember from last time, we can think of DNA in this sort of complicated, write every atom down sense. Okay? But it's simpler to really think about 2 main parts of each DNA molecule. Okay? So you have what I called last time the backbone, which is the sugars and the phosphates. Those don't really contain any information. They're just sort of structure. Right? And then the information is in the bases, which are either one of these 4 letters, A, T, G, or C. Okay? And as you remember from the clip I played from Jim Watson, they fit together nicely, right? So when you have A's pairing with T's, there are these bonds that form that keep them linked together, and G's pairing with C's, and so on. And what was left out of that explanation is that it's not as if you have sort of two people standing up next to each other and linking arms like this, okay? It's actually one of the people is upside down, all right? That is, the two strands in a DNA molecule are parallel, but they're what we call anti-parallel. So one of them is facing this way, and the other one is facing this way, okay? And you can sort of see that just because of the way the backbone looks, okay? You can see that this backbone starts with a phosphate and ends not with a phosphate, okay? And the strand that it's paired with starts with a phosphate up here and ends with not a phosphate, okay? And in the scientific lingo, we call this phosphate end the 5' end, and we call the other end the 3' end. That's not really crucial for you to know, but what I do want you to understand is that there is this directionality to it, that it's sort of two people standing in opposite directions, because all of the enzymes we're going to study that either copy DNA or turn the DNA into information that's read somewhere else in the cell have a directionality to them. They can basically only go one direction on this molecule. And so you can think of these as sort of a two-lane highway or something where all the cars have to go this way or that way, and you can't go backwards in the lane that you're in. Okay? So we've been talking about how DNA is the genetic material and how it is copied, and now it's time for us to discuss what a gene actually is. For this, we have to transport ourselves back almost 100 years before the double helical structure of DNA was discovered, before the Avery experiment that we talked about, before the Griffith experiment, even a few years before Miescher first isolated nuclein, which we now know as DNA. And we have to go all the way back to Gregor Mendel in 1866, when he published his reports of his discovery of the laws of inheritance. You probably have all heard of Gregor Mendel by now. You know that he studied pea plants in his Abbey garden. You probably also know that his discovery was not widely accepted at the time. Important scientists either did not know about it or didn't think it was right. And so his work actually had to be rediscovered at about the turn of the 20th century, the beginning of the 1900s. And it's interesting to think about that in terms of sort of the history of science and how information gets transmitted. It's hard to imagine a discovery being made now that isn't instantly spread all over the internet within seconds. And so it's kind of remarkable to try and go back and think about 34 years before the turn of the 20th century when a discovery was made, and really it didn't get incorporated into general knowledge and acceptance until decades later. So what is it that Mendel actually discovered? We now encapsulate his understanding of the laws of inheritance in two basic laws, which we obviously call Mendels Laws. The first law is the law of segregation. And what this means is that differences in inherited traits are due to different versions of genes, that an organism inherits two copies of each gene, one from each parent, one from the mother, and one from the father. And those retain their integrity. Those two copies exist in the individual, but the way the individual appears is not a reflection necessarily of both genes. That is, if the two copies are different, then the so-called dominant version determines the organism's appearance, whereas the recessive version has no noticeable effect. As I said, these two copies of the gene maintain their integrity, and they do what we call segregate during gamete production. That is, when eggs or sperm are made, one copy of each gene enters each egg or sperm, and the two original copies have an equal probability of ending up in the egg or sperm, and we call that particulate inheritance. That is, the genes don't mix in the organism before sperm and egg are made. They retain their sort of separate existence. And then Mendel's second law is the law of independent assortment, and that is the very simple idea that one gene's inheritance is not affected by another's another gene. So if you recall, what Mendel was studying was things like whether the pea seeds were green or yellow or wrinkled or smooth, and the inheritance of one of those genes, say that affected the color of the seed, had no effect on the inheritance of the wrinkled or smooth related gene. That is, they were independent of one another. And so Mendel essentially got it right. We know there are some sort of nuances and complications that exist that he was not aware of, but for the large amount of what he reported, he was entirely right and very sort of insightful and visionary in his thinking. That said, despite that fact, it took another few decades before his work was really appreciated fully, and it was around the turn of the 1900s when that started happening. Mendel referred to genes not by the word gene, but as factors. And so it wasn't until 1909 that the term gene was coined by Vilhelm Johansen, who is a Danish botanist. And he just used the term gene to refer to Mendel's factors, his units of heredity. He also came up with two other very important terms that we still use today, which are genotype and phenotype. Genotype is the genetic makeup of an individual. We now understand that to mean its entire string of A's, T's, C's, and G's in the DNA alphabet. At the time, obviously, in 1909, that was not how one thought of a genotype, because it was not known that DNA was the hereditary material or that it was made up of A's, T's, C's, and G's. And so the genotype was considered the collection of all factors that could affect observable traits. So the genotype in Mendel's pea plants would be which versions of the wrinkled and smooth gene or the yellow and green gene that a particular pea plant had. The phenotype, on the other hand, describes the observable characteristics of an individual, whether or not they're directly determined by genes. So in the case of Mendel's plants, where there clearly was a connection between genes and observable traits, the phenotypes were yellow seeds or green seeds and wrinkled or smooth seeds. And we now associate particular genotypes with particular phenotypes, and in a sense that's the business of genetics, or the business of genomics, is trying to find associations between the genetic material and how an organism looks or behaves. So we go on from 1909 a few years, and we've already talked about how in 1916 the fly group up at Columbia made a very, very important discovery, which is that genes are carried on chromosomes. And they proved that, as I said in a previous lecture, by watching really rare exceptions to the inheritance rule and using fruit flies, and in particular the eye color of fruit flies. And so if the expected outcome was a fruit fly's eye was supposed to be white and it turned out to be red, Bridges and the other members of Morgan's fly group were able to show that those also were associated with problems with the segregation of the chromosomes inside cells. And so that made a direct connection between genes and chromosomes. And then as we march through the first half of the 20th century, we go from genes are on chromosomes to an understanding of DNA as a chemical substance that is the hereditary material. So the other experiments we talked about, the Griffith experiment, which showed that a chemical substance could actually transform a bacterium to have a new phenotype, changing a rough Streptococcus bacterium into a smooth one. The Avery, McLeod, and McCarty experiment, which we talked about, which demonstrated that that chemical substance was DNA, and it wasn't protein, which everyone thought it would be. So that was, again, a sort of surprising discovery that was not immediately accepted, and we talked about that last time. And then in 1953, we have the discovery of the double helical structure of DNA by Watson and Crick, which we also talked about. So it's interesting then to think about the progression in terms of how scientists and the general public thought about what a gene was, going from Mendel's time up until Watson and Crick. So in Mendel's time, you had this very abstract notion of a unit of heredity, some factor that controlled the observable appearance of organisms. And it was unknown what those factors were made of, how they actually achieved this role of determining the appearance of organisms, but their existence could be inferred by doing crosses, by doing genetic experiments. But as time progressed into the middle of the 20th century, and by the time of Watson and Crick, the way people thought of a gene was in this very molecular, very chemical, very what we call reductionist sense of a stretch of DNA, a string of letters, A's, T's, C's, and G's, that somehow carry information. And what's important to realize, too, is that this was not just a trend that was happening in isolation, but one of the reasons why this idea of information carried in DNA took on such importance and was in the sort of scientific mindset at the time was this was right after World War II. During World War II, code making and code breaking was a very, very important activity. And so it was sort of in the general mindset to think about DNA as a code, the same way we think about messages being sent and interpreted by the right people. So at about that time, right, you now have an appreciation of DNA with a particular structure, with this capacity to carry information, and thinking of it as being a code, and as the task of science from that point on of trying to unravel that code, trying to decipher that code. And what became immediately apparent was that there was some piece of the puzzle that was missing, because you had a paradox, which is that DNA, which is supposed to be carrying this information, is in the nucleus of a cell. And proteins, which do the business of the cell, we've already talked about how complex proteins are and how they're constituents of your cells. They do important jobs, like they act as enzymes to catalyze chemical reactions. Proteins are doing the business of the cell. So it was sort of immediately apparent to people that somehow the information in DNA had to get translated into proteins. But the problem is that the DNA is sitting in one place in the cell and the proteins are made in a different part of the cell. So this is a picture from your textbook. It's a very complicated drawing or rendering of what a cell looks like. And so you can see the purple big blob in the middle with the dimples in it is the nucleus of the cell. It's where the DNA is. You can see the double helix represented in there. Obviously, this is not to scale. The double helix is tiny compared to, or at least its width is tiny compared to the size of the nucleus. And then outside of the nucleus is the cytoplasm, and there's lots and lots going on in the cytoplasm. There are these mitochondria, which are the energy factories of the cell, and we'll talk about those in a later lecture. And most importantly, you can see that there are these thin stretches of membrane that have these little dots on them. Those dots are ribosomes. The ribosomes are the factories that make protein. And so before we talk about the ribosome some more, I think we can do well by simplifying this diagram and taking a look at sort of only the essential information for discussing this problem of how the information in DNA gets translated into protein. So we can take a look at this simpler diagram, which shows many of the same things. You see the nucleus in blue in the center of the cell. You see the mitochondria, which are these elliptical orange bodies, and you see a bunch of other things. Again, you see these membranes extending outside the nucleus that contain these dots on them. That's called the rough endoplasmic reticulum. The dots are the ribosomes where the protein is made. So again, this is even too complicated, and we can do with a much simpler drawing of what the essential information here is that I'm trying to get across. And we can go to a diagram like this. So we have a nucleus in the center of the cell. That's where the DNA is. We have the cytoplasm outside of that. And in the cytoplasm are the ribosomes, and that's where proteins are made. That's where protein synthesis happens. So somehow the information has to get from the DNA in the nucleus to the ribosomes. And so it was postulated that there was a messenger. There was some missing messenger that was taking the information from the DNA to the ribosomes. We now know what that messenger is, and it's a molecule that's chemically related to DNA called RNA. And in fact, because it's a messenger, it's a particular flavor of RNA called mRNA, or messenger RNA. And you can view the clips on the DNAi DVD by these guys who were involved in sort of discovering that connection. So let's talk a little bit more about messenger RNA. RNA is chemically very close to DNA. The main difference is it's a different sugar than DNA. So DNA, remember, is made up of the sugar and phosphate backbone and the nitrogenous bases, which are the A's, C's, G's, and T's. So the sugar in DNA is deoxyribose, and the sugar in RNA is ribose, slightly different sugar. Don't ask me why that is, it just is, and there are lots of cases like this in biology where things just are, and there's not necessarily any good explanation for why there would be a different sugar in RNA than there is in DNA. Also, and this is another example, RNA, instead of using the same letters A, C, G, and T, uses A, C, and G, but it has a base uracil instead of thymine, so it has a U instead of a T. So the same base pairing rules apply. If DNA has an A, the corresponding base in the RNA would be a U instead of a T. If DNA has a C, the corresponding base would be a G. Unlike DNA, messenger RNA is single-stranded. That is, DNA exists in these double-stranded, double helices, so you have A's matched with T's, G's matched with C's. Messenger RNA is only a single strand. It's not matched up with anything. And the role of messenger RNA is to carry the genetic instructions for making proteins. That is, it takes the information from the DNA and brings it to the ribosome. And we're going to talk about how that information actually gets translated. So what we have to think about now is how do you convert the DNA language to the protein language. So DNA and RNA contain 4 letters, the nitrogenous bases, A, C, G, and either T or U. And that has to get converted somehow into proteins. Proteins are made up of amino acids, and that alphabet has 20 letters. Remember, because it had 20 letters, that's why people originally thought that protein was the genetic material, because 20 sounds a lot more complicated than 4, and so people thought the more complicated thing was going to end up being the genetic material, and they were wrong about that. So let's look at these amino acids. They form polymers the same way DNA forms a polymer, of these repeating units that are attached end to end, one after the other. DNA can stretch for many, 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 many letters. Each one of our chromosomes probably has millions of letters. Proteins are generally much shorter than that. The typical protein is a few hundred to maybe 1,000 or 2,000 amino acids long. So this is the basic structure of an amino acid. There's this amino group, the NH2, that gives it its name, and the carboxy group at the other end, the C double bond O, and negatively charged oxygen, which gives it the other part of its name as an acid. These get connected to each other, and the R hanging off the side is the variable part of the amino acid. So the nitrogenous base is the variable part of DNA. It's either A, C, G, or T. The R in the amino acid could be any one of 20 different things. And so here are those 20 things. And you can see the part in gray is the part that's constant for all amino acids, and the part off to the left of each one is the unique part of each amino acid, and they fall into different categories. So some of them, these side chains are themselves acidic or basic. Some of them are so-called aromatic compounds. They have these benzene-like rings in them, and so on. So they fall into different categories. These amino acids give the proteins their structure and their activity. So the exact sequence of amino acids in a protein and where the basic and acidic and neutral and aromatic side chains fall basically makes a protein what it is. You join, as I said, the amino acids one to the next, and that makes a peptide or a protein. So these amino acids are connected one to the next, and so this is how the process goes. You have your first amino acid, and you can think about lining up the next one, and the carboxy end of the first gets attached to the amino end of the second, and a covalent bond is formed between them, and then the carboxy end of the second one is now available to be attached to the amino end of the next one, and so on, and so on, and so on, and you form this peptide chain. So as I've said before, This is not a chemistry class, and I don't expect you to remember these exact chemical structures. I think it's important to know that proteins are made up of repeating units, but that these repeating units come in 20 different varieties. And so the way I think about a peptide chain or a protein is not in terms of these amino and carboxy ends being attached to one another, but in terms of what those little R's are that are hanging off the side. So let's imagine the first one is the side chain that corresponds to the amino acid methionine. So let's replace that R with its three-letter abbreviation, methionine, MET. And let's say the second one is the amino acid alanine, and the third one is the amino acid proline. So we can forget about all of this molecular detail, all this chemistry, and just write it as methionine, alanine, proline. And in fact, we can just write it as regular text as methionine, alanine, proline, right in a row. So that basically is how proteins are put together. But how does the DNA alphabet of 4 letters get converted into the protein alphabet of 20 letters? For that, as you can see, is exactly a code. You can imagine writing something in one alphabet and deciphering it in another alphabet. And so the task then is to figure out what that genetic code is. And a lot of effort took place by a number of scientists to figure out this code. And in the third lab that you're going to do, we simulate that process of discovering what the genetic code is, so you'll get a better understanding of how that was actually done. For now, let's talk about what the properties of that code have to be. And so if you think about converting an alphabet of 4 letters into an alphabet of 20 letters, how might you do that? Clearly, it's not just a matter of replacing one letter with some other letter. Let's say replace A with methionine, replace C with proline, replace G with alanine. You run out of letters, right? You only have 4 letters in DNA, so that allows you to represent at most 4 amino acids if you have a 1-to-1 relationship. So 4 letters into a 1-to-1 code to 4 amino acids is not enough. You only get 4 amino acids that way. So what I want you to think about now is how you would get 20 letters out of 4 letters of DNA, how a code like that might work. So you can think about having 2 letters in DNA correspond to each amino acid, but then how many combinations of 2 letters do you get? Well, you get 4 times 4, or 16 different combinations. You get AA, AT, AC, AG, CA, CT, CC, CG, and so on. That's 16 combinations. So that's not enough to represent 20 letters of amino acids. So if we go to 3 letters of DNA corresponding to 1 letter of amino acid, then that will work, because then we get 4 times 4 times 4, or 64 possible combinations. We get AAA, AAC, ACA, ACG, and so on, 64. If you don't believe me, you can write it out yourself. So that's more than enough, right? We only needed 20, but that gives us enough to, if we wanted to, code for 64 different amino acids. And that's basically the answer. So on the DVD, you can watch the little clip about the triplet code. So 3 letters of DNA corresponding to 1 of protein. That's a triplet. So we call that the triplet code. And there are several interview clips with Marshall Nirenberg, who was one of the people who was very instrumental in figuring out the code. So this is the way we represent the code nowadays. You can see that this table is set up so you have the first letter in the RNA, the second letter, and then each of the possible third letters is written in each entry in the table. So for example, UUU, UUC, UUA, UUG are all in the upper left corner. And so it's very easy to look up a particular triplet and see what amino acid it corresponds to. So for example, if I ask you, what is CAA? Then you look in the second row and the third column, CA, and then you see CAA corresponds to the amino acid glutamine. So there are several features of this table that are important to notice. So the first is that many of the amino acids have more than one triplet that that codes for them. So, for example, the amino acid alanine is coded for by GCU, GCC, GCA, and GCG. We say that the code is redundant because there are 4 different ways of coding for alanine. Some of them only have one. So methionine is only AUG, and methionine turns out to be a very important amino acid because every protein starts with methionine. So every RNA has to have an AUG that corresponds to the start of the protein. You'll notice that there are also 3 elements of the code that do not correspond to amino acids. Those are so-called stops, so UAA, UAG, and UGA. Those stops mean that when the RNA is getting converted into protein and that triplet comes up, the protein is done. So it's released from the ribosome and that's it. That's the end of it. And you can imagine that that's an important part of this code. Otherwise, perhaps the proteins would stop at random places or would only stop at the very end of an RNA, and you wouldn't get the exact protein all the time. So we call these triplets codons. So CAU is a codon for histidine, and the UAA, UAG, UGA are called stop codons, and methionine, in addition AUG being the methionine codon, we also call it the start codon because, as I said, all proteins have to start with methionine. Okay, so now we have this code, and this connection between DNA, messenger RNA, and protein is so important that it's been given this kind of semi-mystical name of the central dogma of molecular biology. And so if we think of molecular biology again as a religion, then this is the trinity that we'd be praying to: DNA, messenger RNA, protein. There's your central dogma of molecular biology. And what I want to show you now is how you sort of go in between these different molecules. So let's imagine that we had a DNA sequence that read AT, GG, CCC, and so on. And I asked you, so that's one strand of the DNA, and I asked you for the other strand of that DNA molecule. Can you give me the other strand of the DNA molecule? Yes, you can, because, as we've said before, in the double helix, A's always match with T's, G's always match with C's. So if I give you AT, GG, CCC, and so on, up through TGA, then you can give me the other strand of DNA. And it's important that when we read the other strand of DNA, we read TCA, TAT, AAT, and not TACC, GGG, and so on. And that's because, as we've talked about, the 2 strands of a DNA molecule are anti-parallel. They have a directionality to them, and we always read DNA molecules in one direction, the so-called 5' to 3' direction. So if the first strand is being read from left to right, then its complementary strand is being read from right to left. ATGGCCC is the top strand, and TCATAT is the bottom strand. Now, if we know that that's the DNA molecule, and I tell you that the messenger RNA is going to be made complementary to the bottom strand, then you can give me what the messenger RNA sequence is exactly, right? Because everywhere the DNA says T, you're going to put an A. Everywhere the DNA says A, you're going to put a U. Everywhere it says C, you're going to put a G. Everywhere it says G, you're going to put a C. Okay, so if you have the DNA, you can give me the messenger RNA. And here we have AUG, GCC, and so on. Once you have the messenger RNA, then you can get what the protein sequence is. So we go triplet by triplet, okay, when we always start with methionine. So the first one here, which I've made it to be that way, is AUG, so that's a methionine. And so we can go right along and look up in that genetic code table what each what each codon corresponds to in terms of the amino acids. So the protein sequence corresponding to this messenger RNA is then methionine, alanine, proline, and so on, and that's directly from the genetic code table. And every exam in the course, you'll get the genetic code table as one of the pages of the exam for your reference, because the genetic code is not something you memorize, it's something you look up when you need it. Just as one small note, because you might see it, often we use this 3-letter code for amino acids in proteins, so MET for methionine, ALA for alanine, and so on. Sometimes you'll see it as a 1-letter code, so each one of the amino acids has a single letter that refers to it. And so the single letter code corresponding to this sequence of amino acids is MAP-NSII. And then the stop codon is represented in the 3-letter code as STP, and in the 1-letter code usually as an asterisk. So there's the central dogma of molecular biology. DNA gets made into messenger RNA. Messenger RNA travels out of the nucleus to the ribosome, where it's made into protein. And protein is the thing that's doing the business of the cell. It's what enzymes are made out of. It's what structural proteins like collagen and keratin, things that make your body, are made of. The process of converting DNA into messenger RNA is called transcription. And that makes sense. So it's basically a copying operation that's being done. So you can imagine copying a DNA text into an mRNA text, then you just have to be careful to convert T's into U's. So that's called transcription. The process of converting the information in messenger RNA into proteins is called translation. And that makes sense also because, as we've been talking about, you're basically translating from one alphabet, from one language, into another alphabet, another language, from the DNA alphabet or the RNA alphabet of 4 letters into the protein alphabet of 20 letters. So there's important things also to point out about this. So I said that if you have one strand of DNA, you can get the other strand. If you have the strand of DNA, you can get the messenger RNA sequence. If you have the messenger RNA sequence, you can get the protein sequence. Now, if I give you the messenger RNA sequence, can you infer what the DNA sequence was? Yes, you can, because again, messenger RNA and DNA have this one-to-one correspondence. If there's an A in the messenger RNA, that corresponds to an AT base pair in the DNA. If you have a U in the messenger RNA, that corresponds to a TA base pair in the DNA. If you have the protein sequence, can you give me the messenger RNA sequence? The answer is not quite, right? So if you know that amino acid methionine is there, then yes, you can give me the exact messenger RNA sequence. It has to be AUG. But if it's most of the other amino acids, for example, alanine, then there are several ways of coding for alanine. There are several codons for alanine. So you can give me that choice, but you can't give me the exact one it is. We don't know if the amino acid is alanine that the RNA was GCC. It could have been something else because there are other ways of coding for alanine. Okay, so let's get back to this question, what is a gene, right? We started way back with Mendel. The gene, he didn't use the word gene, he used the word factor as a unit of heredity. It then became defined in more and more molecular terms as the 1900s proceeded and as a more chemical understanding of the hereditary material came to be. And so a modern definition of a gene, that is a 2010 definition of a gene, is that it's a stretch of DNA corresponding to a messenger RNA. Another way of saying that is it's a transcribed sequence. It's a part of the DNA molecule that will eventually make it into a messenger RNA. And so that's what I've shown here in red on this double helix of DNA, and then the corresponding stretch of messenger RNA, which I'm just drawing as a single line because it's not paired into a double stranded molecule. It's just a single strand. Okay, so that's part of a gene, but it's not the whole gene. So part of the gene is this transcribed sequence that will eventually be made into a messenger RNA. And then another really important part of a gene are associated sequences, which I'm showing here in blue, that control when transcription happens. And so this is a really, really essential point. Every cell in your body has exactly the same DNA in it. So what makes a skin cell different from a liver cell, different from a kidney cell, different from a heart cell, is not what DNA is in it. All of the information is there in every single one of your cells. What makes those cells different from each other, what makes a liver cell produce a lot of enzymes that help with your digestion, what makes a heart cell produce the right materials to make it a functioning muscle and pump, is which genes are transcribed. That is, which stretches of DNA get made into messenger RNA and which messenger RNAs get made into protein. Because what you want to happen in your liver cell, for example, is enzymes to be made. And so the genes that carry the information that codes for those enzyme proteins has to be active. It has to be made into messenger RNA in a liver cell. There are certain digestive enzymes that you would not want to be active in, say, your eye cells or your heart cells, because they could actually do damage. And so in those cells, those genes are not active, they're not transcribed into messenger RNA, and so you don't get those proteins. This is an essential concept in genetics and genomics. All of the DNA is the same in all of your cells, and what makes the cells different is which genes are active, which genes are transcribed into messenger RNA. And so that's a theme that we're going to pick up again in next lecture, and it's going to come up again and again in the course. And actually one of the important ways people do genomics, people study genomes, is by figuring out which messenger RNAs are being made in different cell types, and we'll pick up on that in later lectures.